Hello, everybody. Today we are here for another interview. We are glad to have Julie Graesch from BioWorks Inc. Julie is a biological program manager specialized in insect control management at BioWorks. So, hello, Julie. How are you doing today?
I'm doing very well. Thank you, Sebastian.
So, yeah, before going further in the discussion, can you please give us a quick introduction about you and your background?
Absolutely. So as Sebastian mentioned, I work for BioWorks and excuse me, my position is biological program manager for insects. I have worked for BioWorks for a little over a year. And prior to working at BioWorks, I had 13 years of experience as a biological scientist and entomologist, and I held positions at BASF as a scientist, at Becker Underwood for several different positions, quality control, regulatory, and entomopathogenic nematode specialist, hence why we're here today. And I've also worked for Iowa State University as a research associate. My contact, as long as you can see my screen, my contact information is up, and there's some information. My contact information will be again at the end if you don't get it here. So that's me in a nutshell.
Okay, thank you very much, Julie. And as we saw, like you are specialized in nematodes. So today, maybe I can introduce the topic that will be the nematode usage in greenhouse industry. So first of all, can you maybe tell us a little bit about nematodes, their usage and their application in the greenhouses?
Yes, and I have prepared a few slides because I believe that seeing pictures is a great way to demonstrate the things that I'm talking about. So to answer your question with some slides. So first of all, entomopathogenic nematode. There's actually several different ways that people call nematodes. And by the way, there are many different types of nematodes out there. There are the insect feeding ones, which are the ones that we're talking about. There are plant parasitic nematodes, which are dreaded and well known in our industry and other industries. There's also bacteria feeding, heartworms in dogs and whales and different mammals are caused by nematodes. So there are many, many different types of nematodes out there. Today, we're going to be talking about one specific group called the entomopathogenic nematodes. And entomopathogenic, once again, it simply means insect killing. They're also known by insect parasitic nematodes and beneficial nematodes. So anytime you hear any one of those three comments, they all mean the same thing. They're a good nematode. It's a good guy. So beneficial nematodes are soft-bodied, unsegmented roundworms. They are naturally found in the soil worldwide. They move throughout the soil in response to host cues such as vibrations, carbon dioxide, and other chemicals. And that can be from the insects moving around in the soil. It can be from them respiring the carbon dioxide. It can be from them defecating in the soil. It can also be volatile organic compounds given off by plants as they're fed upon by insects. The plants are saying, help me, I'm being fed upon by an insect. And so many different beneficial organisms use those volatile organic compounds to hone in on a host. Nematodes are no different. So one of the really cool things about beneficial nematodes is they have a really specific life stage called the infective juvenile. And the infective juvenile is the stage that you as a grower will receive when you buy nematodes. 100% will be beneficial infective juveniles, the IJ. These are free living, developmentally arrested, and non-feeding. Okay, what does that mean? So this is the only stage that's found outside of a host. All other life stages, such as adults and eggs and other juvenile stages, are found inside the host. And outside of the host, these infective juveniles are not growing, so they stay as an infective juvenile, and they're not feeding. So the only way they can survive is if they find a host. So just to give you a little idea of what that looks like, here we have you as a grower would purchase the nematodes, mix them in a mix tank, and then spray them out. So this is my sprayer. So you apply the beneficial nematodes to your plants in your greenhouse or nursery or wherever you're applying them to. They can be applied anywhere, and those nematodes will then go in search. Can you see my cursor? Is that moving, Sebastian? Okay, good. So the nematodes are applied to the soil profile or plant foliage, if that's where you choose to apply them, and they go and they seek out, like a snake, snake-like movement through the soil profile to find insect hosts. When they find those insect hosts, they enter through natural body openings such as the mouth, the anus, or spiracles, and they then release a bacteria inside the insect that causes septicemia. And what that is just a fancy name for blood poisoning, just like people can get blood poisoning. So and bacterial infections. So the bacteria then multiply, it kills the host, the nematodes then feed on the host and produce 2 to 3 generations. Again, that's going to be dependent on how large your insect is. The larger the insect, the more generations that the nematodes will produce. Until the insect is being consumed, there's no more resources or they're getting limited, then the adults will then start producing the infective juveniles, which again is that really specific life stage. And that is then what leaves the host once limited resources are there. So that in a nutshell is the life cycle of a beneficial nematode. And that is going to be true for the many different species of beneficial nematodes that are out there. We're going to be mostly talking about 1 or 2 of them in this presentation. So I'm going to start this video. This is a video that I took at when I worked for BASF. So BASF does own this video. But I took this is a time-lapse video. It's about 30 minutes, condensed down to about 30 seconds. And what you're going to see is nematodes, which are the tiny worms you see here. And the insect in the middle is a fungus gnat larvae. So what you're going to see is the nematodes, and I'm going to go ahead and start this, are going to be emptying out of the spaces and concentrating on the host. So you'll see that as it goes. I also want to draw your attention to the back end of the fungus gnat right here. You're going to see a nematode go right in on the backside. So bravo. Now likely there's other infection that's happening here. It's just hard to see, but that one was really easy to see. So then what you see here, for one thing, you can see the movement of beneficial nematodes. This is a gel that I put the nematodes in, and this is a live fungus gnat larvae, and so you can see that. Now, before I was able to take this video, I had to allow the fungus gnat to wear itself out because that would make for a very boring video without a fungus gnat to see. So you can see that a lot of nematodes are also in a line kind of here. And so that is a previous location of that fungus gnat larvae. So the nematodes, once again, are attracted to larvae themselves because they're respiring, they're breathing, they're letting off carbon dioxide, and they're also attracted to previous locations of the insect because they're leaving a trail per se. So this is a really, really cool video. I love showing this video, and it just really illustrates. Now they don't move like that in nature. They're a little slower, but this is a great video. The second video that I want to show you is this one. Again, this is our fungus gnat larvae, and what you see, again, this is a little different. This is real time, and this is just in water. And there's a lot going on, but this is the black head capsule of the fungus gnat larvae. And what you're seeing here is the head of 3 or 4 different beneficial nematodes, and they really want to get into the mouth of this fungus gnat larvae. And this fungus gnat is alive, so it's kind of clenched its mouth closed, and it's really irritated. If we continue watching the whole thing, you can start to see it move a little bit. But I just wanted to show you this, that it illustrates how driven these nematodes are into getting into their host. So what are some advantages to using beneficial nematodes? And this is starting to get at your question. First of all, they have a reliable performance. They've been used for many years, 50 plus. As soon as they were recognized as as an organism that will kill insects, which is pretty easy to see when you have an insect and all these worms are coming out of it. And it's grotesque, but it's also really cool. So it's really easy to see. And so researchers like, oh, what's this? They pick it up. It looks like it killed the insect. Let's see if it'll kill other insects, and away you go. So it's been researched heavily throughout the years. There's a lot of data on the use of beneficial nematodes. Once they get inside the insect and release the bacteria, they typically will kill the insect within 24 to 48 hours. They're suitable for IPM strategies, so they are compatible with many chemistries, and we'll talk about that briefly here in a second. The application strategies are similar to chemicals, and they're applied through standard equipment. Again, this is going to be important for ease of use.
Can I ask you a question about the application? Like,
absolutely.
There are like 2 different ways of application, like the drenching with the water hose, with the, like you put the nematode in a watering tank, and you water with your hose with a high volume of water, or you can also apply it with the sprayer, where there is a little bit higher pressure and less volume per square meters and everything. Which one is your favorite to get the best efficiency?
Thank you for that. And it really somewhat depends on the crop. But if you're targeting things and where the insect is that you're targeting. So if you're targeting something in the soil profile, you may need a heavier volume of water. If you are applying to the foliage, you'd want a lower volume of water. And that is because if you had a higher volume of water, you'd have runoff. And obviously that's if you're targeting the foliage, and you're just going to wash your nematodes off the foliage. One of the things that was a red flag to me with the comment that you made is high pressure. And we're going to talk about high pressure here in a slide or two, but nematodes will not be able to tolerate 300 psi. So if your application equipment is a higher pressure than at 300 or higher, then nematodes aren't going to work in that type of system. And I'm going to tell you why here in a minute. Did I answer your question?
Yes, yes. Go ahead.
So last couple of points here, they are safe for the crop users and the environment. They're extremely little carrier typically in most nematode formulations. So what you're applying is typically pure nematodes, and they can't survive on or in humans or other animals. So they only insects. So they're not regulated by the EPA because they're indigenous and they're safe. So that's the no government requirements. Also, there's no reentry intervals because there's no EPA requirements. So it's very versatile, very easy to use. However, there are some things that we need to know about in order to prevent killing the nematodes during the application. So another thing that we should talk about is when we need to apply beneficial nematodes. Preventative applications are the best. So preventative meaning before insect populations get to the point where they're a nuisance or so high that you're really impacting your plants. If they're eating the roots or eating the foliage, depending on what stage you're controlling. So you need to make sure that you stay on top of your insect populations, monitoring. This is a plug for Sebastian here, monitoring your pest populations by using some sort of tracking software like BugVision. And so that is highly, highly important, not just tracking what is going on and keeping graphs, but you will also know how well your applications are working because you can see your population of insects going up and down based on what you're applying. And in addition, you can also see what you did in the past, what worked, what didn't work, and know when pest populations show up. So highly, highly beneficial and highly recommended. You want to make sure that you're, go ahead, Sebastian.
Knowing that that's mainly helping for the soil and the roots of the plants, like avoiding attacks on the roots and everything. Maybe that's something that people should use anytime right away on the nursery plants, like the one that are rooting, like the cuttings or anything like that, so that they are sure that the rooting is perfect from the very beginning.
Very good point. So if you have a highly susceptible crop, whether that is a crop that is in propagation, so you're either doing that from seed or transplants or sticking, anytime you have, say, a fungus gnat in that type of system, it's going to chew on those newly emerged roots. And if you do that, not only are you going to stop or impede the plant's ability to uptake water and nutrients, you've also introduced a wound where soil pathogens can enter, and that can be a huge concern because there's a lot of soil pathogens out there. So it's very important to start early, preventatively, so that those fungus gnats don't get a hold of and don't gain in numbers and are not only damaging your plants but also causing pathogens, fungal pathogens, bacterial pathogens from causing problems in your plant. So you want to target the correct life stage, and that's really important because if you were to target the adult flying stage of the fungus gnat, that's not the right stage to target with beneficial nematodes. So you have to use something else. And this is what we call the pitchfork approach. So you'd want to use the beneficial nematodes to control populations of the larvae. You could use something like an azadirachtin or an insect growth regulator to help there as well, because anytime and actually that's a really good point to make here, in that know what is compatible with the beneficial nematodes and know what is synergistic. So if you use a low rate of an insect growth regulator, then that is going to slow or stop the molting process of the insect. And when you've done that, it makes the insect sick, if it doesn't outright kill it, that is. So if you've made the insect sick, then the nematodes, so the insect immune system is compromised, and the nematodes have just that much more easy time getting in and overcoming the insect's immune system and taking over. So insect growth regulators are really great products to use with things like beneficial nematodes and entomopathogenic fungus like Beauveria bassiana. So you want to target the correct life stage. Typically that's a soil dwelling stage, but it doesn't always have to be. It can also be different life stages. And if you are curious to know if a certain insect will be killed and what life stage will be killed, you are free to reach out to me if you want, and I will help you answer those questions. The best time to apply beneficial nematodes is going to be early morning or late evening. You can do it during the day if you have something like blackout curtains, where you can pull those curtains, and this just has to be long enough for the beneficial nematodes to be applied to the soil or the foliage. And soil, they have to get down into the soil profile, and if they're applied to the foliage, I would say you would want to leave those blackout curtains on as long as you possibly feel comfortable leaving those blackout curtains out, because they need time to find their host and infect their host before the foliage dries out. Because as soon as the foliage dries out, they have no protection and they will desiccate or dry out and die. So the foliar applications of nematodes is a very, very brief application. And as soon as the crop is dry, the nematodes are gone. So you really have to make sure you're aware of that if you're trying to make an application to the foliage. It can be done. And you want to apply under ideal environmental conditions, which we've already talked about. So as I mentioned a little bit ago, you want to apply through you can apply through commonly used application equipment. However, there are a few things that you need to know in order to prevent killing the nematodes. The first one is you want to agitate to prevent settling. And nematodes will settle out very quickly. So it's very important to have some sort of agitation. Oxygen or an air pump is going to be the best, because you are not only oxygenating the water, but you're also stirring the water. So that's the best. You can also use mechanical pumps, although that can be a little bit harsher on the nematodes, and it can also heat up your water. So you have to be a little careful in that you need to monitor the temperature of your water, especially if it takes several hours to apply a nematode application. You heat up that water and you will kill the nematodes. So it's very important. The nematodes come packaged with cold packs. Throw those in the freezer, and when you make a nematode application, if it's a hot summer day, throw those in your spray tank, and that will help keep the water cool. Cool water also holds more oxygen. Again, another benefit. Beneficial nematodes are not aquatic, so they don't swim, and they won't survive long term in a water solution. So the sooner you can get them applied, the better. You can use adjuvants and wetting agents both to the foliage and to the soil, and that will help things tremendously. If you've ever applied water to a leaf, which I'm sure you all have, you'll notice that without a spray adjuvant, the water droplets will ball up on the leaf, and you've essentially caged the nematodes. So you do, if you're applying to the foliage, need to have some sort of adjuvant or wetting agent to break that surface tension and spread the nematodes out over the foliage. And the same sort of concept in the soil, except you have soil particles. You need free moisture in the soil in order for the nematodes to move around on the soil particles and get through the soil profile. So you need a little bit of free moisture. And if you have basically a surfactant or wetting agents or soap-like, so if you have that in there, it just makes it easier for the nematodes to navigate the soil profile. You want to remove all filters of 50 mesh or finer. Finer filters will start to collect nematodes, and then they will start to push through and shred. So that's obviously a bad thing. Nozzle apertures of smaller than 0.5 millimeters is bad because, well, for one thing, this is mist, and you want larger droplet sizes getting down to either the foliage or into the soil profile. And also that can also cause some shear stress as they go through that tiny nozzle. And so it's just important to use larger apertures so that you get larger droplet sizes. Finally, and we already talked about this last one, don't exceed 300 PSI. They go from very high in the pump system, and then when they're sprayed, they go to a very low pressure or no pressure, and they can actually explode. So also not a good thing to do to a nematode. So here's just some examples of application equipment that can be used with beneficial nematodes. I promised you that we would talk about compatibility. This is extremely important. And fortunately, one of the things that make nematodes great is often they're known as the gateway to biocontrol because they are 100% a live organism, and so considered a BCA or biological control agent. And so that can be scary for growers who have never used live organisms or biological control. But nematodes are surprisingly compatible with many, many different things. And that may surprise you, being that they're so small. However, they're so resilient because of what I talked about earlier with their life cycle. Remember, I said that they're an infective juvenile and they're not developing and they're not eating. And they also have what I usually refer to as a rain jacket, in that they're not a first stage juvenile, they're a second stage juvenile, and they have retained the molt, their previous molt, and so and that is surrounding them. So they have this rain jacket that allows them to be very protected from things like the chemicals. Since they're not eating, they don't need to have access to the outside, outside of the rain jacket. So they're very protected. So that's really cool, really interesting tidbit about beneficial nematodes. So there are things that aren't compatible with nematodes. If you're using beneficial nematodes, whoever your supplier for beneficial nematodes are, they should have a compatibility guide of what is and what is not compatible. There's several guides out there, so I encourage you to look for those. And then again, just for your awareness, things on this side are examples that are chemicals that are not compatible, and the ones over here are examples of things that are compatible. It is also important, and this is my last slide before we actually talk about any specific pests. It's important to check nematode viability. So you as a grower, you want to know that your biological control agents or natural enemies that you receive in are alive. And you can do that very easily with beneficial nematodes. For one thing, make sure your cold pack is cold upon delivery. Make sure you put them into the refrigerator as soon as you receive them. And you can open up a very tiny corner of the pack, remove a very tiny amount of the formulation, put a piece of tape on that if you're not planning on using it immediately, just to make sure that you don't let any moisture out, because there's not very much moisture in there. It's very precise. So if you leave it open, like if you want to use half a pack, and you just throw the other half back into your refrigerator, which we don't recommend, by the way, all of that moisture in a very quick amount of time will evaporate, and you kill the rest of the pack. So that's one reason. But to get back to my point, you can grab a little bit from the pack, put it in under a microscope, or if you have a hand lens, you can see the nematodes moving around. It's easier to see with a microscope, very easy to tell live from dead nematodes. So viable, not viable. If you see the pin straight, if you see pencils, something went wrong. So you don't want to see that. It's also really great to check nematode viability, not just when you receive them, but let's say you ordered too much and you kept it for a couple months. Are those still alive? Well, the shelf life says it should be, but how can you be sure? Check it. How can you be sure that they survived going through your application equipment? Check it. Put out a tray of or a bowl or whatever in your greenhouse when you make the application, collect some of the water, and look at the nematodes when they come out the other end of the hose. And that's just a really quick and easy way that you can see whether or not they are surviving the application. Also important on hot days. So there's many reasons that you should check viability or check them all. And it's very easy to tell, and it's a very quick thing. So it shouldn't take too much time from your growers. So that's my intro crash course to beneficial nematodes. The rest of the slides are based on specific pests, fungus gnats, thrips. What do you have any questions for me, Sebastian, up until this point?
No, I was listening carefully because that's really good and detailed information, and I think the listeners will learn a lot. Like I learned a lot from this presentation already, and yeah, I think we can keep going on the specific pest. Maybe you can like we talked a lot about fungus gnats and fungus gnat larvas. Like, can you give us more information maybe on thrips? Because maybe that's the kind of like nematodes against thrips is kind of controversial in the industry. So what is your opinion, and do you think it really works on thrips?
Thank you for that question, Sebastian, and great question. So I'm gonna skip through the there we go, thrips. So thrips are tiny. And in fact, they're really not that much bigger than the nematode itself. So it is a difficult application. And if you're going to target adults on the foliage, which you can, like I said, that is a more difficult application. And there has to be a lot of thought and care put into that type of application. If you're interested in that, call me and I can help you walk through it. The easier application would be applications made to the soil. And so what's important to know about Western flower thrips, and I'm mentioning Western flower thrips here because they're very common thrip. There are other thrips out there, like onion thrips, that are, I think, are rising in onion and chili thrips, that are rising in recognition. So you may think everything is a Western flower thrip. No, that's not necessarily the case. And actually, Vineland in Canada is actually working on a onion thrip study to see what the actual distribution of onion thrips is from Western flower thrips. So it's a really interesting study, and actually BioWorks is one of the companies that's helping out fund that trial. So one of the things that makes nematodes possible for Western flower thrip control is that there is a life stage that goes to the soil. Now, I want to also mention here that not all thrips pupate in the soil. There is the echinothrips. They don't go to the soil. So if you are familiar with the life cycle of a thrip, you have an egg, you have several instars, you have a prepupae still on the plant, then you have a pupa, which interestingly, if you're familiar with insect biology, this is a really interesting pest. most pupae are immobile, completely 100. I can say that with fairly high certainty. There's probably like 99.9% of insects don't have mobile pupal stages. Thrips do. Now, they don't move around very much, and really probably only if you poke and prod them, but they can move. They still have legs, they still can move. But those will fall off the plant and fall into the soil, and voila, you now have a stage in which beneficial nematodes can kill. So that's the stage that we're going after. So we're going to target the pupal stage with nematodes, but remember, we still have the adults and the larval stages that are on your plant causing damage. So you need to have other biological agents that are going to, if you're using a biological program, you need to have other BCAs going after the adults or eggs. There's a lot of really great ones out there for thrips, or you need to have some sort of IPM strategy to combat the other stages. And this is about the size of a nematode in comparison to a thrip. Just again for size comparison. So the reason why I'm telling you all of this about the pupae is another very good point to make is that if you have flowers, and thrips love flowers because they eat pollen, and if you have pollen available for thrips, their fecundity or their ability to produce offspring will skyrocket. So once you have pollen available for the thrip, you really need to have your control agents in place before that happens. Otherwise, things can get out of hand extremely quickly with thrips. Also, once you have flowers, most of your thrips will probably migrate to the flowers. And flowers, especially dense flowers like chrysanthemums or gerbera daisies, they're facing up. And those thrips, even if they're pupating, can actually still pupate in that flower. And that is an opportunity for a foliar application because water is going to stay in the flower longer. So this is a good, if you have flowers, if it's just foliage, it's tricky. If you have flowers, it's a little less tricky because they do hold water longer. So as you can see with this rose, there's very little, there's less likelihood, as you can see from flowers and no flowers, thrips are pupating in the soil, regardless of if there's a flower there or not. However, like I said with the chrysanthemum, there's a much higher likelihood that during flowering stage of, say, a chrysanthemum, many more of the thrips pupae will pupate on the plant. This is just an example of why, again, you should use adjuvants. And we talked about that earlier. Sebastian mentioned the drench application versus the foliar applications. Again, if you're going to apply to the foliage, if there's flowers, you need to let that flower stay wet for at least 2 hours. Things you can do is turn blackout curtains, turn off artificial lighting, close vents. So once again, here is a good example of the size of a beneficial nematode compared to, and this is actually a pupae. And the reason I can tell that this is a pupae is because the antennae of the pupae are folded back on top of the thrip's head. So this pupae was killed by a beneficial nematode because it was dissected and out pops the nematode. So, and then this is a dead adult and Steiner Nemafeltii. This is an example of a study that was done for Western flower thrip activity. And the reason I'm bringing this up is the reason why it's important to apply nematodes in the early morning or the late evening, because your thrip activity is going to be, the thrips are going to be most active in the day. So if you apply in the afternoon or evening, especially if, and this goes for soil or foliar, but especially foliar, you don't want an active thrip. So this is just an example of why it's important to apply nighttime and in the morning. This is a study that was done, again, by BASF. This was done, I believe, I should have had that on there, but at Vineland again. And here we have percent control on the vertical axis. We have the blue bar is a chemical standard, and then the red bar is our nematodes. So the percent control isn't the same to start off with, because those beneficial nematodes, they need to go through the soil profile to find those thrips. And so it takes a little bit of time, but, and by the way, there was weekly treatments of beneficial nematodes at 7, 14, 21, 28, and 36. And assessments obviously are made all along the way as well. So you can see that with a single application of your chemical, that you can, and weekly applications of nematodes, you can get to the same level of control, or close to the same level of control as a chemical. And as we all probably also know, thrips are extremely susceptible to resistance in chemicals. And so so you have to be really careful not to over apply chemicals, because you don't want the insects to become resistant to your chemicals, because then you become severely limited in what you can apply. So here's another one done by Vineland. Did you have a question, Sebastian?
Yeah, I just wanted to summarize. So the rules are start early, because as you're, it's mainly working on pupa. So you got also already the adults, so you got to work on the young generations of thrips, and also being consistent, because we see that the efficiency rise after so many weeks. So you don't want to find to see a result right away. If you do like this, you will just stop, and that's just when it starts working. So
that's an excellent point, and thank you, Sebastian. You truly have to. Nematodes are not a one and done application like a chemical. You do have to make regular applications of beneficial nematodes. Because it's a live organism, and this is an inundative release, meaning you put out lots of nematodes to try to control the population, and 98% of those are probably going to die because heat and temperature. But when you put them out at that high of number, then they're more likely to find a host. Anything that doesn't find a host is going to die in, probably, depending on temperature, there's a lot of variables here, but under ideal circumstances, those beneficial nematodes could probably survive a week or two, just depending on environmental conditions, temperature, heat, humidity, UV, so on and so forth. So here is another study done by Vineland. Here we have the control, which is the blue bar that has more exponential population growth, and this is the number of western flower thrips. Then we have two other bars here. We have a drench foliar and a sprench. And a sprench is just basically a foliar application applied to runoff. So that runoff then goes into the soil. So you're sort of getting the best of both worlds. And so here you can see both do a really nice job of keeping the population in check. And the last application was probably made right here, either week 8 or week 9. And so then you can see in the absence of the nematode, the constant nematode applications, which again here were applied weekly, the population immediately starts to climb. So you do have, especially with the thrip, and especially when there's pollen, you have to keep on top of those. And that's, so we do have a product called Nemashield, and we do have a lot of information. We have the available sizes, your rates, shelf life, and so on and so forth. Any nematode company is going to have something similar. And with that, I'm going to open it up to Sebastian to ask me any other questions that I haven't covered.
Sorry, I have to switch it on. No, it's great. Thank you very much for this presentation. I think lots of people will learn a lot from it, and maybe they will start using it more in their greenhouse. I think it's a really good way and a good tool in the BCA toolbox to control the pests in a greenhouse, and also thinking about using it like the sprayer. The person that's spraying is not taking any risk spraying nematodes, so it doesn't matter if you got to apply it every week. Like, it's not like chemicals where they have to wear the mask and they have to take precautions to make sure that they don't get like they don't get in contact with the product. So, and yeah, like I think to sum up is more like everything needs to be consistent, and it's not only about nematodes. It can be for any kind of biocontrol agent. Like, you got to start and be consistent in your application. That's the only way to make it work, and that's the only way to be successful. So thank you very much, Julie. Well, I will provide again your contact information at the end of the video so that people can reach you.
I might have.
And
go back up to the top.
Yeah, on the first one. Yeah. So I will anyway copy it at the end of the interview. And yeah, thank you very much for participating to that interview. I think that was really interesting and a lot of information. And thank you again.
You are most welcome, Sebastian, and thank you for having me on. I really appreciate it.
Yeah, thank you very much, Julie. Have a great afternoon.
You too.